Hi, I'm Erin Piazza. I'm a senior bioinformatics scientist at NanoString. My role at NanoString is to lead content development from the bioinformatics side across our various platforms. The most recent panel I've worked on is called the nCounter ADC Development Panel. My Ph.D. was actually in cancer biology and tubulin function and tubulin-based structures. So it's actually been a pleasure to work on a panel that so closely aligns with some of my old research interests. To explain antibody-drug conjugates, we need to think about the current paradigm of cancer care. Cancer therapeutics for millennia have included surgery, dating back to the ancient Egyptians. However, in the last few hundred years, we've developed radiotherapy and then cytotoxic chemotherapy, particularly in the wake of World War II. Much more recently, however, we've moved into the era of molecular therapeutics and now immunotherapeutics. While we certainly have some newer, more modern therapies that we can use against cancer, it's quite common for standard of care to still include cytotoxic chemotherapy for many tumor types. While cytotoxic chemotherapy is nonspecific, typically dosage is used as a mechanism to try to target tumors as opposed to normal cells. That being said, we know that there's still quite a bit of systemic toxicity from using cytotoxic chemotherapy. It's into the context of cytotoxic chemotherapy and its issues that we introduced the antibody-drug conjugate. The goal of ADCs is to overcome the lack of specificity offered by cytotoxic chemotherapy. In short, we use an antibody to specifically target tumor cells as opposed to normal cells instead of just dosage alone. This also offers us the opportunity to use far more powerful cytotoxic agents, in this case we call them payloads, to target tumors, hopefully offering something that has far less resistance mechanisms or possibility than standard cytotoxic chemotherapy. Over the years at NanoString, we've developed a broad variety of oncology panels, some with rather broad motivations, including our IO360 panel that thinks globally about the tumor microenvironment, and the immune response, and then some more focused panels that think about particular applications, like our CAR-T characterization panel. However, we're constantly talking to people in the field and creating new relationships with different researchers, and it's in the last few years that we have developed some relationships with people who work in the area of ADC therapeutics. This piqued our interest in this particular topic, and we learned with time that while we had some panels with overlap with research interests in this area, we didn't have a single offering that really gave researchers what they needed for this particular therapeutic type of research. And so it's for this reason that we determined we would develop an ADC development panel. The panel thinks about ADCs in the context of a typical life cycle of ADC. I say typical because there are some more modern approaches that don't necessarily have a similar life cycle, particularly as it relates to internalization of the ADC. But in short, most ADCs start with binding of the ADC to the target on the tumor cell itself. The ADC is then internalized by receptor-mediated endocytosis and brought into the cell. That endosome then matures into a lysosome, and through a variety of possible mechanisms, the payload is released from the antibody in the process of lysosome maturation and function. This can be done by pH, by specific enzymes, or by degradation of the antibody itself. This then leaves the payload free to exit the lysosome when it degrades and enter the cytoplasm, where it can perform its particular mechanism of action. Typically, these mechanisms of action include something related to microtubules or possibly to DNA damage, amongst other very core cellular functions. Ideally, this is intended to kill the tumor cell, but sometimes we know that payloads actually escape the cell via drug efflux pumps, which is also a common mechanism of resistance in cytotoxic chemotherapy. We also know that in many cases, the immune response is critically important here and stands sort of outside of this particular diagram, but always related to what we think about in cancer care. The The immune system can be important because ADCs are combined with IO therapeutics. It could be that the payload itself is immunological in origin and triggers some sort of an immune response, either an innate immune response, ideally, or in some cases adaptive immune responses. And finally, we know that the cell could die in a way that is immunogenic, causing immunogenic cell death, and finally, an adaptive immune response. The panel we've developed focuses on key questions in ADC research. The first is whether or not the antigen is in fact on the target cell, what level that antigen is expressed on target cells, and whether or not the antigen is specific to tumors over normal cells, which is a key component of ADC specificity. Next, we ask questions about the internalization of the ADC itself, whether or not the ADC was successfully internalized, as this is crucial to ADC function. Next, we ask about payload release. Specifically, did we achieve linker separation, and what possible enzymes could catalyze payload release, as we know that while there are certain design requests or intentions for linker release, sometimes many different enzymes could catalyze a linker cleavage, and it's important to know which of many enzymes are present in any particular patient. Next, for drug MOA and toxicity, we want to know if there are aspects of tumor biology that could modulate or perhaps prohibit the mechanism of action intended by the payload. Further, what is the state of the immune system, and is it primed to respond to cells that might die in an immunogenic fashion? Further, what is the state of various pathways for cell death in the tumor, and are these pathways primed to respond to the mechanism of the payload? And finally, mechanisms of resistance. Are there specific drug efflux pumps in particular that could enable escape of the payload from the cell? The nCounter ADC Development Panel contains 770 genes that enable you to ask deep biological questions relevant to each stage of ADC development. These include tumor targeting and antigen expression, ADC internalization, payload release, drug MOA and toxicity, target cell death, mechanisms of resistance, and finally, the role of the immune response in all of this. This enables you to address biological function with deep molecular characterization in a way that can complement or especially extend beyond standard cell-based assays. This panel is compatible with many different cell types, including cell lines, xenografts, and tissue samples. We also provide an option that enables you to quantify the relative abundance of 14 different immune cell types in a method that we published previously and use across a number of different panels in our portfolio. And finally, this comes with all the benefits of the nCounter system, which includes a simple sample to answer in 24 hours with just as little as 30 minutes of hands-on time. Thank you for your attention.